Welcome my all fellow GATE aspirants to another important topic video from your syllabus. Yes, this is again from section 2, which is biochemistry part. Today we are going to talk about carbohydrates. I've chosen this particular topic because you do get sometimes questions regarding its stereoisomerism and regarding its structural properties also. So let's dig in without any further delay. The first point is about the structure. What exactly are carbohydrates? Yes, they are organic compounds which are consisting of carbon, hydrogen and oxygen. These three type of atoms will be there in carbohydrates, and because its name is carbohydrates, so they are hydrates of carbon, which means for every carbon which is present, there is a water molecule for it. So for every carbon is in the hydrated form. That's why they are called as carbohydrates. The general formula is CnH2n. These are the most abundant biomolecules of Earth. The most diverse biomolecules on Earth will be of proteins, which you will find many different types, but carbohydrates are the ones which are more abundant in nature. They are polyhydroxyaldehydes or ketones with multiple hydroxyl group. Yes, apart from every carbon with a water molecule, you also have some functional groups. The sugar molecules, the carbohydrates which have aldehydes, they are called as aldose sugar. The ones which have ketone, they have keto sugar. They will be called as keto sugar. So over here, either any molecules which already contain those molecules or the substances that yield such compounds on hydrolysis, they are called as carbohydrates. Carbohydrates are building blocks of all, the carbohydrate building blocks are simple sugars which are called as monosaccharides. Mono means single and saccharide means sugar, single sugar molecule. As I mentioned, a monosaccharide can be either a polyhydroxyaldehyde, that is called as an aldose, or polyhydroxyketone, which is called as ketose. So for example, if you have a pentose sugar, let's say, pentose means 5 carbon molecules are there, 5 carbon atoms are there. Now if it is an aldose sugar, the keto carbon will be in the first carbon if it is an aldose sugar, and the hydrogen will be attached over here. So this is what is the functional group, aldehyde functional group. So every sugar will have in either one of that, and then the rest of them, they will be balanced by water molecule. So the arrangement can be different types of arrangement can be there. Okay, and here also there will be water, but additionally one more hydrogen will be there. So we will have CH2OH. So this is what is a ribose sugar or a pentose sugar, but we will also call it as a aldose sugar. Okay, and if we have a keto sugar, then I'll be drawing same a 5 carbon sugar. The keto entity will be in the second carbon, and then the rest of it will be as it is. So this will be CH2OH. These all will be having the CHOH parts, and even this last one will be CH2OH. This is what is the difference between a keto sugar, and what is the difference between an aldehyde group. This is what is called as the ketone group. Okay, that was about the structure of the carbohydrate. Let's move on to the general properties. So sugars can be structurally represented in any of these three forms. So either it can be an open chain structure, hemiacetal structure, or Howard structure. Let's have a look at all this. Open chain structure means it's simple. The form which I have just drawn on the screen, those are long straight chain form of carbohydrates. Yes, they are linear form, straight chain. And then you have the formation of hemiacetal. So let's have a look how this hemiacetal is formed. This is the linear form, which is the open chain structure, but the sugars will generally tend to be present in this cyclic form. This is what is referred to as the cyclic form of the molecule. So here we have first of all, this entire molecule, this will have some chiral carbon. What do you refer as chiral carbon? The one which is composed of a chiral carbon which is having all 4 groups attached to it will be different functional groups. So all the groups attached to this carbon, if they are different functional groups, then that carbon is called as chiral carbon. If that is the case, then this chiral carbon will be having some other forms or some other arrangements. Okay, so those arrangements, they are called as stereoisomers. If you have one chiral carbon, you will have two stereoisomers possible. Okay, I'm coming to that a little later. Now here, whatever is the functional group entity, that carbon is referred to as the first carbon. And the chiral carbon which is farthest away from the functional group. So here first carbon is the functional group, and the carbon which is farthest from the functional group, chiral carbon, those two will interact with each other. Now see, this is written in a twisted form. As you can see, the first carbon is here, and then the rest of the molecule is written like this. Rest of the molecule is written like this. Okay. Placing the fifth carbon here and the first carbon here. Now there will be some interaction. This lone pair of electrons on the OH group, they will target the carbon. Okay. And the oxygen will form a bond with the carbon. So here you can see they have formed a bond. Right. And this carbon, it already had 4 bonds. But if it forms one bond with oxygen, then it breaks one bond. So instead of double bond O, it forms a single bond O. But oxygen needs to fulfill its valency, so it bonds with a H. Now that's why you can see this first carbon, initially it was not chiral, as the oxygen was involved in double bond. But now this carbon got a chirality, additional chirality. This carbon is what is called as anomeric carbon. Okay, this carbon is called as anomeric carbon. Now, what happened in this cyclization, this entire process cyclization? The interaction between an aldehyde group and an alcohol group. So as you can see here, there is an aldehyde group, there is an alcohol group. This gives rise to a hemiacetal structure. So here, whatever structure it has formed here, this structure is what is a hemiacetal. But this hemiacetal has another alcohol group. So hemiacetal can further interact with another alcohol group to further give a acetal group. Now that will happen when you have some sugars. Let's say you have some sugar over here. You have the OH group, and then you have some other sugar. So that also has some OH group. So if this OH and H, this is what? This is a hemiacetal. And what is this? This is another alcohol group, right? So these two will interact together and water molecule will come out from here. A water molecule will be lost. So what will it give rise to in that case? Yes, you will have the sugars which are joined by oxygen, right? So what is this formed between these two sugars? What is this bond which is formed? It is called as glycosidic bond. Yes, glycosidic bond formation is a condensation reaction where water molecule is lost. But actually it is the interaction specifically between a hemiacetal group and an alcohol group. The other alcohol group doesn't always need to be from a sugar. The hemiacetal group should be there, one anomeric carbon should be involved, but the other group doesn't need to be OH, doesn't need to be from a sugar. So it's a misconception that glycosidic bond can be formed only between two sugars. No, one sugar anomeric carbon is required, another can be any other molecule with a OH group. So how is this hemiacetal structure formed? First carbon of the glucose condenses with the OH group of the fifth carbon to form a ring-like structure. For example, this was given for D-glucose, okay, and the other one, so what type of sugar is this? This is an aldose sugar. And the example of a keto sugar is a fructose molecule. This is also a 6-carbon sugar, so it's a hexose sugar, but instead of aldose functional entity, it has a keto functional entity. So it is a keto 6-carbon sugar. Now for ketos, the interaction is slightly different. So here, the fifth carbon, which is the chiral carbon farthest away from the functional group, it doesn't interact with the first carbon, rather it interacts with the second carbon, which has the keto carbon, it will interact with that group, okay. Logic is same, the lone pair of electrons on this oxygen will target over here, so because of which oxygen and carbon will form a bond together, okay. But when it forms a structure like this, you will notice that the first carbon along with the sixth carbon, they are outside the ring. for this kind of cyclization. Whereas in this kind of cyclization, only 6th carbon is outside the ring. The rest all carbons are inside the ring. So this cyclic structure, this is not aromatic, this is just a cyclic structure. This cyclic structure is formed with 1 oxygen and 5 carbon. So this particular type of molecule, certain type of ring structure is what is called as a pyran ring, consisting of 6-membered ring in which 1 is oxygen, another 5 is carbon. And here you will notice that 1 is oxygen and another 4 carbon is present. Such kind of a structure is called as a furan ring. Now that's why what is the proper name for this? It is a D sugar. As we know, carbohydrates are mostly present in the form of D sugar. I was talking about the chiral carbons in sugar. So they can have different stereoisomers which are enantiomers named as D and L. The D sugars are more prevalent. That's why the name is D-fructofuranose, because it has come from a D-fructose and the type of ring that it has formed is a furan ring. So while this is the linear projection and this is the Haworth projection, that's what this is about, Haworth structure. It is basically the presence of the pyranose or the furanose ring-like structures. Now let's go to the physical properties. So physical properties, yes, we'll be discussing about the stereoisomerism. To some extent, I already mentioned to you that these are the compounds which have some structural formula, same structural formula, but they differ in their spatial configuration. Yes, so if you have a chiral carbon, then you will have another form of it. Glucose, for example, it has 2 isomers with respect to penultimate carbon atom. Now, penultimate means the carbon atom which is farthest away from its functional entity. Now, for example, here I have taken glucose. Now, here how many carbons are chiral? This carbon is chiral. Why? Because this is different group, this is different group, this is different group, and this entire thing is a different group. As I mentioned, a chiral carbon, they will be having 4 different functional groups attached. Okay. So don't just see the immediate atom connected. You have to see what is connected overall to the functional groups, what are connected to carbon. Okay. This is chiral, this is chiral, this is chiral, this is chiral. This is not chiral because it has double bond with the same atom. This is not chiral because it has 2 atoms which are same. Okay. That is also not chiral. Now, since this 6-carbon sugar has 4 chiral atoms, 4 chiral carbons, the formula to calculate the number of stereoisomers is 2 to the power n, where n stands for how many chiral is there. That's why for this 6-carbon sugar, the formula is 2 to the power 4, which means that it will have 16 stereoisomers. So yes, for 6-carbon sugars, you actually have 16 different isomers, 16 different arrangements in space, 16 different spatial configuration. Out of those 16, 2 are here. That is the glucose in the D form and glucose in the L form. Like this, you will have mannose, D form, L form. You'll have galactose, glucose, idulose. So like this, different types of 6-carbon sugars are there. Glucose is just one of them. Now, glucose itself, there can be different arrangements in this chiral carbon. OH can be this side or OH can be this side. But on what basis will I call it D or L? So basically, you have to see the penultimate carbon. Penultimate carbon means the carbon, chiral carbon which is farthest away from the functional group. So here, this particular carbon, you have to see whether OH is present on the right. If OH is present on the right side, keeping the functional group at top, then it is a type of D-glucose. If OH is present on the left-hand side of the penultimate carbon, chiral carbon, then it is called as L-glucose. By the way, these two, they are called as enantiomers. Okay. They are also called as enantiomers and also they are optical isomer. So why enantiomer? It means non-superimposable mirror images of each other. So if you look at it as if you have placed a mirror in between them, this structure looks a mirror image of this one. Okay. And what are optical isomers? Because they will reflect change the direction of light in 2 different directions. If D-glucose, if you pass light, wavelength of light from D-glucose, it will bend it to one direction. If you pass through L-glucose, it will bend it to completely different direction, but opposite direction. That's why they are called as the optical isomers. Okay. So as I mentioned, remember, for carbohydrates, the main type of isomer which you see is D type. Okay. Now what are enantiomers? As I mentioned, they are stereoisomers. Enantiomers are type of stereoisomers which are non-superimposable mirror images of each other. The L and D forms of sugar, which is D-glyceraldehyde and L-glyceraldehyde, based on that, these are 3 carbon sugars. What I gave you example was 6 carbon sugar. And optical activity, yes, they will have rotation of plane polarized light forming plus glucose and minus glucose. So basically the D-glucose is also called as plus glucose. L-glucose is also called as minus glucose. Next are diastereoisomers. As I mentioned that the chiral carbons in this hexose sugar, there will be total 16. Out of those 16, 2 are glucose. Now, there can be some other 6 carbon sugars. So one of them is mannose. You will have D-mannose and L-mannose. Now question is D-mannose and D-glucose. They are both 6 carbon sugars. But what are they to each other? Are they mirror images? No. Because D-glucose already has a mirror image, that is L-glucose. So how can it have 2 mirror images? So that means what? D-glucose and D-mannose, they have the same chiral carbon. So what does that mean? Are they stereoisomers? Yes, they are stereoisomers. But are they mirror images? No. So are they enantiomer? No, they are not enantiomer. Okay. But they are non-mirror images. So non-mirror images which are come from the same stereoisomers, they are called as diastereoisomers. Okay. They are non-mirror images. The conformational changes can be in the other carbons because we are not talking about penultimate carbon. If you talk about D-glucose and D-mannose, for both of them, the D nature is decided based on the penultimate carbon, whether OH is on the right hand side or not. That means, what will be the difference in D-glucose and D-mannose? It will be in either of these carbons. These arrangements of H and OH will be different. That makes them different in their structure. Okay, so the spatial arrangement will be different, although the chemical formula will be same. That's why they are non-mirror images. Example, mannose and galactose. Next concept is about epimers. So epimers are 2 sugars which differ only in the configuration around 1 carbon atom. Okay, so I was giving you the idea that you can have D-glucose and D-mannose with a difference in 3 carbons. Here, carbon differences can be there. But if it is only between 1 carbon, then it will be called as epimers. Okay. So for example, if you talk about D-mannose and D-glucose, the difference between D-mannose and D-glucose is only the configuration around carbon 2. That is this carbon. So how will it look, the D-glucose structure and the D-mannose structure, if I have to draw? So rest of the things will remain the same. For example, you have the aldehyde functionality group. Then the second carbon arrangement is different. So instead of OH being here, OH will be here and H will be here. Rest all will be same. Okay, because it is configuration is different only around carbon 2. So rest all structures, they will be same as the previous group. Okay, so this is what is your mannose structure. You can see these are both D sugars. Okay, this is same, but this is what is different. So that's what they are. They are not mirror images, but their conformation differs is only 1 carbon. So they are diastereomers also, they are epimers also. Okay, same thing happens with galactose and glucose. So if you take galactose, the configuration will be different around carbon 4. That means if instead of glucose, if you take galactose, the configuration will be different around carbon 4. So instead of OH being here, OH will be here and H will be here. That will be the only difference. Okay. But galactose and mannose, they won't be epimers. Why? Because the configuration will be different around carbon 2 also, carbon 4 also. For mannose, the arrangement is here. For galactose, the arrangement is here. So they will be differing in 2 carbon. So while galactose and mannose, they might be diastereomers, but they are not epimers. Okay. And another one is anomers. So what are anomers? As I mentioned that yes, when you have a cyclic structure, you will notice that there is a possibility where the OH group, the newly formed OH group, it can be present compared to this 6 carbon, it can be present opposite to that orientation. If they are opposite, then it is called as alpha anomeric carbon. Okay, the arrangement is alpha anomer. But it is possible that OH can be here and H can be here. So in that case, the 6th carbon OH and this 1st carbon OH are on the same plane. That type of anomer is called as a beta anomer. As you can see here, they are in the same opposite plane. That's why it is a alpha anomer or alpha sugar. So what are anomerism? Spatial configuration with respect to C1 in aldose and C2 in ketose. The C1 anomeric carbon and the C2, whether you take the keto carbons. Okay. So OH of C5 and OH of anomeric carbon on opposite side, that will be alpha. OH of C5 and OH of anomeric carbon on same side, that will be beta anomer. These are the different type of isomers which are coming under physical properties. You may get questions regarding these in GATE exam. Last is chemical property. So we have some chemical reactions, ozazone formation, Benedict's test, oxidation and reduction to alcohols. So what is ozazone formation? Carbohydrate derivatives when sugars are reacted with excess of phenylhydrazine. So in that case, you can have the formation of glucosazone. Benedict's test, reducing sugars. Reducing sugars means the one which have a free anomeric carbon. That means the C1 carbon is free to react. That is called as reducing sugar. When they are heated in the presence of an alkali, they get converted to reducing species known as indiols. When Benedict's reagent, solution and reducing sugars are heated together, the color change will be there. So it will change to orange red or brick red. And this you can measure spectrophotometrically. If your color change is more, that means the concentration of reducing sugar is more. These are tests for measuring the concentration of reducing sugar. Next is oxidation. So yes, as I mentioned, in these Benedict's test, D-glucose is oxidized to D-gluconic acid. So one of the agents in Benedict's test is copper. So when you're using copper sulfate, the reagent is blue in color. But when copper, it gets oxidized, it gets reduced from 2 plus to 1 plus, that is by gain of electrons. Now who gives these electrons? These carbohydrates. The reducing sugars, they reduce the copper in Benedict's reagent, because of which the blue color converts into red color. That's why we can say, okay, glucose is considered a reducing agent, because when you treat it with Benedict's reagent, the glucose is converted into the form of gluconic acid. That's why glucose is considered a reducing sugar. And the last one is reduction to alcohols. Yes, the CO groups in the open chain forms of carbohydrate, that is this group, it can be reduced to alcohol forms by sodium borohydride or catalytic hydrogenation. And the products thus that you get is alditols, in which there is no aldehyde group, but rather you will have a alcohol group. So that's what Biotecnika students, that was about an important topic of carbohydrates. To try to remember all these points, it will be beneficial for your GATE exam. Thank you very much.